Hello everyone. So in the previous discussions we have classified the epithelium tissues on the basis of the structure and function they perform and the shape of the cells. In this today's discussion we are going to take one of the topic and one of the classification of simple epithelium, that is the simple squamous, and another is the simple cuboid. We are going to take one of them by one. So firstly we have the simple squamous. So let's see them. As mentioned here, single layered cells present and they are flat nucleus with a flat cell. As the diagram is representing, they have a flat cells with a flat nucleus, and the single layered cells are compactly arranged with very little intercellular spaces and matrix. So they have a tiles like view. So suppose you are walking on a footpath and just imagine this kind of the tissues are arranged. So they are very similar with the footpath bricks. So that's why because of the tiles-like view, they are termed as pavement epithelium. Pavement simple squamous epithelium. Next thing is that they have an irregular boundary. Their boundaries are not fixed. No, they are not in their boundary is not in a cuboidal form or in the tall cylinder form or in a polyhedral ending shape. So they have irregular boundaries. Due to irregular boundaries, they are termed as tessellated. Simple squamous epithelium. Next point we have their function is filtration and diffusion. Due to they are of very thin lining, so they can easily perform the function of the diffusion or the filtration. Due to the diffusion, they also known as the to form diffusion boundary. Let's talk about the location of them. So they are found in the first location where they are found is Bowman's capsule. Bowman's capsule is something like that shape of found in the kidney. That part is basically where the glomerulus or the bunch of the vessels passes. Next location we have is endothelium. Endothelium is basically the inner lining of the blood vessels. So they can easily diffuse the particles. Next location is that they have location in air sacs, all the alveoli. So they can easily form the diffusion boundaries. That's why they are also found in this air sacs or the alveoli. Next and the last location I think is going to be the mesothelium. So these are about the locations. So let's discuss about the simple cuboidal. In the simple cuboidal, we found out the cube-like cells with the centered nucleus, as the figure is depicted that they have a centered nuclei with the cube-like cells. They are also known as the germinal epithelium, and that is a very important question by the point of view of me. Why? Because found in gonads. Now what is this gonads? So gonads are basically the testes and ovary. They both are the reproductive parts of the both humans part, that is female and male. So what is the function of them here? They forms they are basically the egg and sperm. Divides. They basically divides to form the egg and sperms there. That's why they are also termed as the germinal epithelium. Next, we are going to discuss about the function of them. So their function is absorption, secretion, and excretion. Let's discuss the location of them, where they are found in. So they are found in DCT. What is DCT? DCT is actually the tubular part of uh, kidney, or B state nephron. Then the acini of pancreas, then in sweat glands. There are some more examples we can write, like the thyroid follicle, another very good example is the ciliary body of our eyes, another very good example is choroid, and ciliary body of eye, choroid, iris is also part of the eye. So these are all the locations where they are found. When we have some modifications here, what is meaning of this term modification? These tissues or cells are modified into another kind of cells or tissues. So in the breast border, they are actually a part of this simple cuboidal, but they have developed microvilli. Now what is this microvilli? Protoplasmic projection, and basically they are non-motile. Means there is no scope of movement in them. So their shape could be like that. We can draw the shape of the brush bordered epithelium here. We can draw there. So this is basically called as the brush bordered or the microvilli. They basically increase the absorptive surface. So they are known as the brush bordered. Here in the brush border they found out in the PCT, which is the proximal convoluted tubule, and this is the distal convoluted tubule. We will write them later. This is proximal convoluted tubule, and DCT is distal convoluted tubule. Next we have ciliated. There is the presence of cilia. Cilia moves. Let's draw here. These projections are termed as cilia. They are actually movable. So we have a very good example of ciliar movement is that the collecting ducts. Ducts of glands. So this is something about the simple cuboidal. We have learned today about this simple squamous and simple cuboidal. So I think this is enough for today. So let's end our discussion over here. Till then, sign off. Happy learning. Enjoying your learning. Thank you.